Hello and welcome everybody to my channel ominibioentrance, and today we will be discussing CSIR PYQs of the year 2017. I will be discussing the applied biology and methodology section. If you are new to my channel, please do like, share and subscribe. Coming to the next year, June 2017. First question. A T0 transgenic plant containing a transgene for herbicide resistance shows two bands on southern blot analysis using a probe that is internal to the restriction sites used for genomic DNA digestion. However, it segregates in a 3:1 ratio for herbicide resistance is to sensitivity in the T1 progeny obtained by self-pollination. Which one of the following statement is correct? So, a T0 transgenic plant, it shows two bands on southern blot analysis, and it segregates in a 3:1 ratio for herbicide resistance and sensitivity in the T1 progeny obtained by self-pollination. Which of the following statement is correct? The T0 plant is a single copy event. It is a double copy event. You got two bands by southern blot. So first option is not correct. The T0 plant is a double copy event and the two transgene copies are tightly linked. They are not tightly linked. They are on two different chromosomes. If it was linked, then we would have got a 1:1 ratio. But in the question it is said that they have 3:1 ratio. So they are integrated on two different chromosomes. So they are not tightly linked. Then third option, the T0 plant is a double copy event and the two transgene copies are integrated in two different chromosomes. 3 is correct answer. So the answer here is option 3. Then next one. In order to check whether a protein has been phosphorylated during treatment with drug, you would perform. So southern hybridization is for DNA analysis. Western blot analysis, that would be correct. CHIP assay is for chromatin immunoprecipitation assay. That also is not correct, and RFLP is also not correct. So the answer here is Western blot analysis. Then next one. Alpha bungarotoxin binds to acetylcholine receptor protein with high specificity and prevents the ion channel opening. This interaction can be exploited to purify acetylcholine receptor from membrane using ion exchange. No. Gel filtration. No, they have not given the molecular weight. Affinity chromatography. Yes, because it is a pure ligand receptor binding. So affinity chromatography can be used here to purify acetylcholine receptor. Then next one. Somatic cell hybridization is used to assign a gene to a particular chromosome. When two cell lines from two different species are fused, they form a heterokaryon which tend to lose chromosome as they divide, preferentially from one species. A panel of cell lines was created from mouse-monkey somatic cell fusions. Each line was examined for the presence of monkey chromosomes and for the production of a given enzyme. The following results are obtained. So the enzyme is present in A, B, D and F. Now see for this pattern, that is plus plus minus plus minus plus in the monkey chromosome. So it is matching with, if you see carefully, you can see that it is matching with 7. 7 also, A is plus, B is plus, C is minus, D is plus, E is minus and F is plus. So the chromosome that has the gene that codes for the given enzyme is on chromosome 7. Answer is option 2. Then next one. Given below are 4 statements regarding genetic transformation of plants in the laboratory. Which of the combinations of the above statements are correct? Plants incapable of sexual reproduction cannot be transformed by agrobacterium tumefaciens. That is not correct. They can be transformed also. Then integration of transgene in organelle R, that is chloroplast genome, occurs primarily by homologous recombination. That is correct. Then an enhancer trap construct used in agrobacterium mediated transformation would contain a functional coding sequence of a reporter gene and a minimal promoter. That is also correct. A T0 transgenic plant containing two unlinked copies of a selection marker gene HPT and one copy of the passenger gene GFP could segregate in a 1:1 ratio for hygromycin resistance to sensitivity in the back-crossed progeny grown on selection media. It is not 1:1 ratio, it is 3:1 ratio. So option D is not correct. So the answer here is option B and C. Then next one. Agrobacterium mediated transformation was used to generate transgenic plants using a construct with a selection marker gene X and a passenger gene Y. Expression levels of Y protein in 8 independent transgenic plants are given below. The following could represent probable reasons for the observed variability in the transgene expression level. Which of the following combinations of the above statement is correct? Now, position effect on passenger gene. That can be one reason. Then transgene silencing of the marker gene. They have told only on the expression of Y and not X. So X here is marker gene. So B is not correct answer. Variation in copy number of passenger gene. More copy number means more expression. So answer C is also correct. Then mRNA instability of marker gene. That is also not correct because I already told you that they have told only about the expression levels of Y protein, not of X. So option D is also not correct. So the answer here is A and C. Option 1. The next one. In an experiment designed to clone PCR amplified fragments in a cloning vector digested with XHO and SMA1. So XHO is a sticky end cutter and SMA1 is a blunt end cutter. Which one of the following combination of restriction enzymes can be used in the PCR primer to generate compatible ends for cloning? This mark, that is a slash mark, indicates the site of cleavage within the recognition sequence. So here SMA1 is a blunt end cutter and XHO1 is a sticky end cutter. In case of blunt end cutting, any end could be compatible with any end and hence you can use any enzyme which is a blunt end cutter for your PCR primer. Then first choice, that is XBA1 and SPE1 is ruled out because both are sticky end cutters. So the second enzyme should be a blunt end cutter. So there are 3 other options where second site is a blunt end cutter, but we have to see for XHO1 which region is compatible to XHO1. Which region is compatible to XHO1? So if you see here, that is, this is the XHO1 sequence and this is the SMA1 sequence. If you cut XHO1, you will get GAGCT as the overhang end. Now you have to see out of these restriction sites which on cutting will give an end in such a way that it is compatible to AGCT region of the XHO1 digest. Now SMA1 is a blunt end cutter, so I told you that any other end which gives a blunt end cutting can be used, any enzyme. But here it should be compatible. So XHO1, we found that if you use the option 3, that is SAL1 and EcoR5. This is SAL1. SAL1 upon cutting you will get TCGA as your overhang end, 3' overhang end. This is your 3' XHO1 cutting is giving you a 3' overhang end, and this one is giving you 5' overhang end. So upon ligating these two, which are compatible to each other, or complementary base pairing is there. See here, this is XHO1, this is GCT, and this one TCGAC, that is compatible to this end of XHO1. SAL1 overhang is complementary to XHO1 overhang. So it can be used. SAL1 can be used. Here anything can be used because it is just a blunt end cutting. So the option here is option 3, that is SAL1 and ECO R5. Then next one. A researcher was working with 3 proteins A, B, C, which may have potential role in gene expression. In order to validate the hypothesis, EMSA, electrophoretic mobility shift assay, was performed. The purified proteins were allowed to bind with a labeled DNA, and the results obtained after autoradiography are shown below. Now EMSA is conducted to see for DNA protein interactions. So in this question, the researcher was working with proteins A, B and C, and it may have role in gene expression, they say. So in order to validate the hypothesis, they conducted EMSA to see for protein DNA interaction, and the autoradiography that was obtained was shown in the figure here. The following interpretations were made. Choose the correct combination of interpretation. So if you see the first one, no proteins A, B, C is added, no band, only the control band is present. Then this one, if option B is added, no band. If C is added, also no band. But if A is added, you got a band. You got a band. If you add A and B, you got 2 bands. If you add B and C, no band. If you add A and C, only 1 band corresponding to A. Okay. Now you can see from, we will see the statement interpretations. Protein A possesses a DNA binding motif. If you, that is correct, because here the 1, 2, 3, 4th lane, if you see, if when A is added, there you got a band over there. But if you add B, there is no band. If you add C also, there is no band. So protein A possesses the DNA binding motif. That is correct. Protein B possesses DNA binding motif. No, it doesn't have as per lane 2. Then protein B binds to DNA protein A complex. That could be correct, because when you see this lane, 1, 2, 3, 4, 5th lane, you can see that you got a band for A also as well as for B also. So there is only when A bind, B can bind. But if B alone is present, it cannot bind. That is why you didn't get a band when B alone was added in 2nd lane. So option 3 is also correct. Protein C binds to DNA only when protein A is bound. Protein C doesn't bind to DNA even when A is bound as per the last lane. A is also present, C is also present, but you got band for A alone. So option 4 is not correct. So the answer here is 1 and 3. Option 2. 1 is also correct, 3 is also correct. Then next one. Which of the following next month, that is December 2017. Which of the following host system is best suited to express large amount of glycosylated protein for structural studies? So glycosylated proteins, you get it usually for mammalian cells or eukaryotic cells. So it is better if you go for baculovirus, native protein in baculovirus. Answer is option 3. Then which of the following statements about yaks is false? Yaks can carry DNA fragments of 1 MB. That is correct. A single yeast may contain more than one yak. That is also correct. Rearrangements in yaks are rare because recombination is poor in yeast. That is not correct. Yaks recombination is also not that poor in yeast. Rearrangements are also possible in yeast. Then yaks have to be transferred to bacteria for subsequent DNA manipulation. That is also correct. So option 3 is not correct. Then next one. Transgenic tobacco plant overexpressing isopentenyl transferase transferase under the control of promoter region of senescence associated receptor kinase were exposed to drought for 15 days followed by rewatering for 7 days. The following hypotheses were proposed regarding changes in the transgenic plants at the end of 7 days of rewatering. So IPT gene, this was almost a repeated question, but there was difference in the promoter sequence. It was asked somewhere around 15 or some year 15, I think. I'm not sure. But anyway, I have answered one question based on, which is similar to this one. So IPT gene, which is for cytokinin biosynthesis. So reduction in cytokinin transport from roots to shoots allows senescence to occur. But exogenous application of cytosine, it retards senescence to detached leaves. Now you have a construct which is where IPT gene is overexpressing under the control of PSARK, that is senescence associated receptor kinase promoter. Now PSARK is activated only at the onset of senescence. This results in cytokinin biosynthesis in the leaves which inhibit leaf senescence. So the plant would be healthy and would show higher production of cytokinin compared to the wild type plant. So the answer here is option 3, that is B and C are correct. B is the plant would be healthy and survive, and C is the plant would show higher production of cytokinin compared to wild type plants. So both options, that is B and C, are correct. Then some of the following transgenic approaches could be used for functional characterization of endogenous genes in plants. Which of the following combinations can be used? Transformation using a binary vector containing a strong enhancer element and lacking right border of tDNA. That is not possible. Then transformation using binary vector containing a promoterless reporter gene sequence and a selection marker gene cassette within the tDNA. That could be used. Transformation using binary vector containing only a strong enhancer element and a selection marker gene cassette within the tDNA. That is also correct. Then next one, transformation using a binary vector lacking a reporter gene as well as both the left and the right borders. So D is also not correct. So option B and C are the only statements which are correct. So answer is option 2. Then next one, match the following Vir proteins with their correct function during agrobacterium mediated transfer of tDNA to plant cells. So Vir G is for induction of Vir genes, that is option 4 and not option 4, fourth statement. Then Vir D2 is nucleus targeting of tDNA 1. Vir B1 is component of membrane structure transfer apparatus for tDNA transfer. Vir F is proteasome mediated destruction of protein coating tDNA complex. So the answer here is option 2, A for B1, C2 and D3. Then next one, match the technique with its appropriate application or use from the list of options given below. CHIP, CHIP, chromatin immunoprecipitation assay. That is for identification of binding sites of transcription factors. So A is 3. Then bisulfite sequencing. Bisulfite sequencing is analysis of methylation site. Option 1. Not option 1. Yes, as per option 1, B is 1. So that can be correct. That we will see for others also. Then ELISA. ELISA is quantification of transgene expression. That's also correct. RLM race. RLM race is for identification of the 5' and 3' ends of transcript. Answer is option 1. Then next one. A researcher attempted to clone a 630 base pair coding sequence of a gene downstream to bacterial promoter 500 base pair in length for overexpression and purification of the encoded protein. The gene sequence was isolated as a SMA1 fragment and cloned at a SMA1 site located downstream to the promoter. The gene sequence contained a single site for EcoR1 located 30 base pair downstream to the start codon. A schematic representation of the plasmid along with the locations of some restriction enzyme sites of the vector is given below. The researcher screened the obtained colonies by double digestion using HIN3 and EcoR1. Which one of the following restriction digestion patterns would represent the restriction profile of the desired clone that could be used for overexpression? So here I have drawn a map of whatever data they have given in the based on the data that has been given in the question. So a researcher attempted to clone 630 base pair coding sequence of a gene downstream to a bacterial promoter 500 base pair in length. So this is the promoter, it is 500 base pair in length. This is the gene of interest, it is 630 base pair. And purification of the encoder. The gene sequence was isolated as a SMA1 fragment. Recognition sequence indicates an arrow. Indicate the site and cloned at the SMA1 site located downstream to the promoter. That is over here. It was cloned at the SMA1 site. The gene was inserted over here, downstream of the promoter. The gene sequence contained a single site for EcoR1 located 30 base pair downstream to the start codon. So this region, this much, that is your 30 base pair site which is having an EcoR1 restriction site. A schematic representation of the plasmid along with the locations of the same restriction enzyme sites of the vector are given below. So they have drawn a schematic representation. The researcher screened the obtained colonies by a double digestion using HIN3 and EcoR1. So the researcher has screened the obtained colonies by double digestion using HIN3 and EcoR1. Which one of the following restriction digestion patterns would represent the restriction profile of the desired clone that could be used for overexpression? So which of the following is the restriction profile for the clone which is obtained by double digestion with HIN3 and EcoR1? So this is your map. Now this is your HIN3 site and this is your EcoR1. Now if you digest it with HIN3 and EcoR1, you will of course you will get a 500 plus this 30 base pair sequence will give you 530 base pair sequence, and the, will be this one will be one of the fragments that you will obtain. Then you have got EcoR1, there are two sites, so this region will also be cut off. So here the remaining will be, after this region has been removed, the remaining will be 600 base pair plus this 30 base pair. So that will be around 630 base pair. Sorry, that will be around 630 base pair after EcoR1 digestion. So this is the second band, and then you will get your vector backbone. So you will get as per option 1 530 base pair plus approximately 600 base pair plus vector backbone. So answer will be option 1. The next one, diagnosis of influenza virus infection can be done using some of the following techniques. Western blot, southern blot. Now, first of all, influenza is a single-stranded RNA virus. So, southern blot is not possible. Northern blot, western blot. That could be one method. Northern blot is for RNA and western blot is for protein. ELISA and RT-PCR. That is also correct for influenza virus infection diagnosis. PCR and electron microscopy. No. So choose the combination of techniques that correctly list the detection, that is B and C. Answer is option 3.